If you wouldn't mind, just type your first name or your nickname into chat for me. Looks like just about everybody is here. Maybe one or two more. It's good to see everybody. Today, we're going to be talking a little bit more about carbohydrate catabolism. We're going to be discussing fermentation. So we've mentioned fermentation a lot this week, but we haven't really talked about how we test for it. Remember, when we do an OF test, what we're really asking is an either or. Can the organism perform carbohydrate catabolism through oxidative means or fermentative means or both? E. coli was an example of an organism that can do both. But sometimes what we want to do once we know that an organism is fermentative is we want to ask, what kind of carbohydrates can it ferment? The truth is that all living cells, as we've said in here, are capable of using glucose in order to produce energy, in order to produce ATP. But there are some microbes who are capable of using carbohydrates other than glucose in order to obtain glucose. Remember, we said when we were talking about carbohydrate terminology, we said that there are lots of disaccharide molecules that are combined simple sugars that are bound together. For example, we said lactose, that's milk sugar, is simply one molecule of glucose and one molecule of galactose that are bound together. So there are some microbes who can ferment lactose. And what they're doing first is breaking the lactose down into those two simple sugars. And then they take the glucose and they ferment it. Similarly, a sugar like maltose, which is just two glucose molecules bound together. There are some microbes that have the enzyme needed to break apart or catabolize that molecule into two glucoses. And then it uses that glucose for fermentation. So different microbes, depending on the species, can use certain disaccharides in order to obtain glucose. And they need the glucose, of course, in order to make their ATP. And if they're doing it through fermentation, we can test for that. So we've already looked at the OF test again, which is sort of the can they ferment test. And what we'll do today is we'll look to see a little more specifically about what they're fermenting. In other words, what molecule are they using to get the glucose to ferment it? So that's what our objective is today. First thing I want to do is ask if anybody has any questions about what we talked about last time, or any questions about anything that you've been looking at or reading, quizzes, anything like that. Feel free to type your question in the chat, and we can certainly talk about stuff. Otherwise, what I'll do is I'll just jump right into our slides for today, and we'll do a little background first about fermentation. So that's what you should be seeing right now on your screen, is the slide set about the fermentation lab. You should see the title slide on your screen right now. it's really important when we talk about metabolic processes that we understand what each process is about. And I've been talking this week about 3 primary pathways for catabolizing carbohydrate. Fermentation is its own particular pathway, and we define it as being an anaerobic process that converts the product of glycolysis, which is pyruvate, to some combination of product molecules, alcohol, acid, and or gas. And remember, the microbe is doing fermentation for a reason. The primary reason is to make ATP. Sometimes we're looking at microbes that are using fermentation to produce ATP when their primary pathway is not available to them. So when cellular respiration is not possible. Maybe they're an aerobic cellular respiration organism and there's no oxygen around. Or maybe they're an anaerobic cellular respiration organism and their final electron acceptor molecule is just not available. And so they switch to a secondary metabolic pathway in fermentation. Still, other organisms are going to use fermentation just as a second primary pathway. There are microbes that use fermentation equally well with cellular respiration. Neither pathway is preferable to the other. So they just switch back and forth. And still other microbes, as we mentioned last time, are what we call pure fermenters. They only use fermentation to make their ATP. Those are using fermentation as their primary metabolic pathway, which is kind of fascinating. So some organisms are using oxidative catabolism. They're using cellular respiration. And what follows cellular respiration, which is called oxidative phosphorylation. In order to oxidize or break down or catabolize, those all mean the same thing, glucose, in order to make ATP. Organisms that do cellular respiration with oxygen are the aerobes. The final electron acceptor is oxygen. Organisms that don't use oxygen are the anaerobes. They're doing respiration with nitrate or sulfate or carbonate as their final electron acceptor. The key thing to remember is this. Just because fermentation is anaerobic does not mean it's the same pathway as anaerobic cellular respiration. Those are 2 separate pathways. You've heard me say it before. You'll probably hear me say it again. They are 2 completely separate pathways. Anaerobic cellular respiration looks almost identical to the aerobic process. The only difference is that final electron acceptor. In an anaerobe, they're going to use nitrate or sulfate or carbonate at the end of their electron transport chain. Whereas you and I and other aerobes are going to use oxygen. Fermentation is completely separate from that. Fermentation is its own pathway. All cells, remember, can break glucose apart in order to make ATP. All cells can do it on this planet. All the different microbes can do it. All of the multicellular organisms can do it. It's just that we all do it a little differently. Microbes are capable of a much broader range of metabolism than we are. They are extremely diverse in their metabolic activities. And so when we're trying to identify a microbe, we can test all of its metabolic abilities. And we can use that data to identify that microbe. We can take all those test results and we can go to a textbook or another source and we can look up what organism has all of these things as characteristics. What organism, what's their gram status? What happens when you try to put them on MSA? What happens when you put them on EMB? What happens when you put them on McConkey's agar? What happens when you put them in an OF tube? What happens when you put them on a starch plate? You gather all that data and then you go to source material to find out which organism has the same characteristics as your unknown. And that's how you identify what you have in the microbiology lab, anyway. Now I have a little note down at the bottom here. The beginning part of fermentation is identical to the beginning part of cellular respiration, and that is the series of reactions called glycolysis. Glycolysis is a series of reactions. In humans and other aerobes, it's a 10-step reaction, and it takes a glucose molecule and it basically splits it into two, into what we call pyruvate. Now, in fermentation, glycolysis still occurs. It's just that the pyruvate is going to be sent through fermentation and not through respiration like it normally would. So it turns out that microbes, because they're so diverse, can sometimes do glycolysis just a little bit differently from one another. They use a slightly different combination of reactions than what humans use. So I put together a little supplement for you about glycolysis in microbes, and it's available to you on Canvas. You don't have to read it. Don't feel compelled to read it. But if you're interested, it's about a one-page summary of some of the differences that we see among microbes regarding this process called glycolysis. So when we talk about cellular respiration, we're talking about glycolysis, followed by pyruvate processing, followed by the Krebs cycle. That is the series of reactions that is going to take a glucose molecule and break it apart and set all of its electrons free. During glycolysis, the glucose is broken down into this molecule called pyruvate. This part of cellular respiration, this glycolysis, does not require any oxygen. This is why glycolysis and fermentation are anaerobic. You don't have to have oxygen around to do glycolysis. During fermentation, you still get the pyruvate made. So glycolysis goes on, you get your pyruvate, but instead of proceeding into pyruvate processing and then the Krebs cycle, what happens is the pyruvate gets sent down another pathway. And this is a pathway that's going to produce, it's going to break apart the pyruvate, and it's going to produce ATP for that microbe, as well as some product molecules. The pyruvate is going to become something else. Sometimes it becomes an acid, sometimes an alcohol, sometimes a gas, and you see combinations of these. So to the microbe, fermentation is about producing ATP. It also does something else, though, for the microbe. It helps regenerate some of these electron carrier molecules that I mentioned in lecture. Remember, as the glucose molecule gets broken down and all the electrons get released, we have to have molecules around that can hold on to those electrons temporarily and then give them or donate them to the electron transport chain. These electron carrier molecules have to be available or else you can't do cellular respiration. So one of the things fermentation does is regenerate or replenish the stores of electron carrier molecules. So that if you're an organism that would really rather be doing respiration, but you can't do it right now because you don't have your final electron acceptor available, so you're fermenting. Well, by regenerating those carriers, you're going to be able to jump back into cellular respiration as soon as the conditions improve. So the number one reason that a microbe ferments is to make ATP. But number two, it also helps get ready to do oxidative catabolism again as soon as the environment improves. Fermentation is not an efficient way to make ATP. That's just the truth. For the same amount of glucose, if I start with the same amount of glucose, I'm going to get a lot more ATP made if I do cellular respiration than if I do fermentation. So just in terms of efficiency, aerobic cellular respiration and anaerobic cellular respiration are much better methods than fermentation. And that's because of the literal way that they make the ATP. In cellular respiration, the last part of the process involves the energy from those electrons that were taken out of the glucose being used to build ATP in a process called oxidative phosphorylation. Very, very efficient process. In fermentation, instead, we see something called substrate level phosphorylation. You're still making ATP, but you're doing it differently. You're doing it in a much less efficient way, too. You're not going to get as much ATP built from the same amount of glucose. This little picture over here, I showed in the lecture about fermentation. This green glob represents an enzyme. You can see it's got these divots in it. These are active sites in the enzyme. I know it's hard to see, but there's a little white bar here. That represents just some kind of substrate molecule, something that's going to be turned into a product with the help of this enzyme. So this molecule could be any of a number of things that inside the microbial cell is just in the process of being turned into a product molecule. But here's what makes it interesting. There's a secondary active site where ADP binds. So as this white bar, which has a little yellow circle on it, which represents a phosphate group, as this white bar gets converted into a product molecule, and that's what they're showing us over here, the phosphate group gets taken off of the substrate and added on to the ADP. So by the end of the reaction, you've got your product and you've got ATP made along the way. We often refer to this as piggybacking. The production of this ATP was just piggybacked onto the production of this white product molecule. So it works, it makes ATP, it'll keep an organism alive. It's just not very efficient. Substrate level phosphorylation just isn't very efficient. Lots and lots of different carbohydrates can be fermented by different species of microbes. Every cell can do glucose, but some microbes can get that glucose from another carbohydrate source. So some can take lactose and break it down to get glucose. Some can take sucrose and break it down to get glucose. fructose, mannitol. There's lots and lots of sugars that different microbes can ferment. So the key thing is that depending on the species of bacteria, they may or may not be able to make use of these sugars in fermentation. You have to have the enzyme necessary. You have to have the enzyme necessary, for example, to take a lactose molecule. And remember, lactose is just one glucose plus one galactose that are bound together. You have to have the enzyme to break that bond between those two molecules and set the glucose free. Because then once you have that glucose, you can turn it into pyruvate through fermentation. So we can use fermentation abilities to help identify microbes. And there are lots of fermentation pathways. If you look into textbooks, there are lots of different fermentation pathways depending on the species. Bacteria ferment, fungi ferment, archaea ferment. Lots of organisms have this ability. But the two most common fermentation pathways are definitely the lactic acid pathway and the ethanol pathway. So remember, in lactic acid fermentation, and this is the one that human cells can do, remember, the glucose molecule first goes through glycolysis, because that always happens, and becomes pyruvate. And then the pyruvate is converted into lactic acid and ATP, again, through that substrate level phosphorylation. In ethanol fermentation, there's a little extra step in there. It's not quite as simple, but it's close. It's close. Glucose gets converted to pyruvate. Then the pyruvate gets converted into a molecule called acetaldehyde plus a molecule of carbon dioxide. And then the acetaldehyde gets converted into ethanol. And that's when the ATP gets made. So these are the two most common pathways you'll see, but there are many others. And remember, we take advantage of fermentation. We humans, we take advantage of the fact that microbes can ferment. We do it on an industrial scale. We grow microbes on an industrial scale, and we push them to ferment because the fermentation process is going to make products that we find valuable. The alcohols, the acids, the gases, we can harvest those things from microbes as they are being produced, and they have commercial value. So yes, ethanol is certainly a valuable molecule produced by yeast primarily through fermentation. But in addition to ethanol, there are other alcohols. Butanol is made through fermentation. Butanediol is made through fermentation. These are commercially valuable alcohols. In the acid department, I mentioned lactic acid. There's commercial applications for this in food production. Butyric acid, formic acid, succinic acid, propionic acid. There are lots of different acids that we can now make commercially by allowing microbes to do fermentation on an industrial scale. We can harvest the acids and we can use them, sell them commercially. The two most common gases that are made through fermentation are carbon dioxide and hydrogen gas. And again, they have value. This is a chart that comes right out of your textbook. These are some of the more common fermentation pathways. So again, as I mentioned, lactic acid is in this list. So is ethanol, or alcohol, as they have it called. You can see what they've done is they've named the pathway over here. Then they list the end products that get made from the glucose and the pyruvate. Then they give you some example organisms that can do this kind of fermentation. And then they tell you what the commercial product is. So a lot of food stuff, a lot of foods come from fermentation processes, but also other things, pharmaceuticals, cosmetics, solvents, things like that. So we have gotten very good on an industrial scale at growing microbes. If you ever go into one of these facilities, they have gigantic vats that are full of growth medium, culture medium. And they optimize the culture medium to grow whatever microbe they're trying to grow. And then they just make sure that the environment in that gigantic culture container is going to push the microbe to ferment. And then once certain amount of time has gone by, they can basically drain out the fluid and they can distill out the product. So that's what happens all over the country, all over the world, in commercial facilities, industrial facilities that are producing fermentation products. It's just microbiology on a massive scale. All right. Does anybody have any questions about what I've been saying so far? Does it make sense to you? We like fermentation because we take advantage of fermentation, right? Human beings like it when microbes ferment because we can harvest the products that they're making. But the microbe is doing the fermentation to make ATP and to replenish the stores of those electron carriers that it can use if it switches back to respiration. Different microbes do fermentation differently. They do all different kinds of fermentation pathways. The most common ones are lactic acid and ethanol, but there are many others. And what kind of fermentation a microbe can do is one of its characteristics. So when we're trying to identify a microbe, we can test its fermentation abilities. The OF test tells us whether they can ferment, but we can do other tests to see what they can ferment. And that's what we'll talk about today. So on this slide, I've listed 3 tests that we'll be looking at to investigate fermentation abilities in microbes. The first one is called the Carbohydrate Fermentation Test. This one has a great name because it tells you exactly what it's about. This test is going to determine if a microbe can ferment a specific kind of sugar. So yes, you could put glucose into this test. If you hadn't already run an OF test, you could certainly run the carbohydrate fermentation test in its place and just put glucose in the tube and see if the organism can ferment it. But you can also put all different other kinds of sugars in there. So if I want to know, for example, let's say I have an intestinal bacterium and I want to know if it's a coliform. I think it might be E. coli. So I want to know if it's a coliform. I'm going to look to see if it can ferment lactose because all coliforms can ferment lactose. I can choose, in other words, which simple sugar I put into the carbohydrate fermentation test. Now, the other thing that this test allows us to look for is the production of acid and the production of gas. We'll know if acid is produced because we'll see a color change in the tube. And we're going to have a little clever way to know whether or not gas is being produced as well. Now, the second kind of test we're going to use today is called the methyl red Voges Proskauer test, and we shorthand this as the Mrvp test. These are actually 2 separate tests, but they are run together because both tests are going to help us determine what kind of acid is being produced during fermentation. The carbohydrate fermentation test is going to let us know if an acid product is being made, because a color change will occur. We can also use the methyl red Voges-Proskauer to tell us what kind of acid is being made. And we do that by pH changes. So not just an acidic pH, but very specific acidic pHs will occur depending upon the acid product that's being produced through fermentation. We can distinguish between an acid that has a pH less than 4.4. We can distinguish an acid that has a pH between 4.4 and 6. And we can find acids that have a pH greater than 6. We can also, with this test, learn if a microbe has the ability to take this acid, whatever it is, and turn it into a product called acetoin. This is a neutral byproduct molecule. And we'll look at this test. And finally on our list today is what's called the citrate test. This is an interesting test. This one looks at whether or not a microbe can ferment a molecule called citrate. Now, what's interesting about this test is this is not a carbohydrate. Citrate is not a carbohydrate. And yet we're going to see if an organism can ferment it. So it is possible to ferment things other than carbohydrates, and citrate is a good example. Citrate is a molecule that shows up in metabolic pathways. It shows up as an intermediate molecule in several different metabolic pathways, including the Krebs cycle. Remember, citrate, you could also call citric acid. And it's a molecule that, again, we see again and again in various metabolic pathways. Whether or not you can ferment citrate is a different question. This is a test that uses an indicator dye, so we're going to see a color change if an organism can take this citrate and make an acid or an alcohol or a gas from it. All right, let's take a look at our organisms for our experiment today. We have our old friend E. coli, of course. We have an organism called Enterobacter aerogenes. This is another gram-negative rod and another coliform, an intestinal organism that can ferment lactose, just like E. coli can. And we have a third very similar organism called Klebsiella Pneumoniae. Now, Klebsiella is an interesting one because while there are lots of strains of Klebsiella that are perfectly harmless, there are some pathogenic strains that can cause very severe disease. Klebsiella, like these other two, is a gram-negative rod, and it's an intestinal organism. It's a fecal organism, and it's a coliform. But if you get a pathogenic strain of Klebsiella pneumoniae, it can cause meningitis, it can cause pneumonia, as the name suggests, and it can cause cellulitis, which is a very serious skin infection. So some strains of this, just like all of these organisms, some strains are pathogenic. Now, those are the first three that we'll look at. We also have two other organisms to consider today. You'll notice that they're also gram-negative rods, and they're also intestinal or fecal organisms, but these two are not coliforms. So they cannot ferment lactose. This is an important distinction that we sometimes have to make, right? If we get a sample into the lab from a patient that has a severe intestinal infection, we know that there's a long list, unfortunately, of bacteria that could be causing it. So one way to differentiate between those bacteria is by figuring out whether or not this disease-causing, this pathogenic bacteria can ferment lactose. If it can, it's a coliform. If it can't, then it belongs to one of these other groups. But identification of intestinal organisms can be tricky because there's a pretty long list of them, unfortunately. So first on that list of the non-coliforms is Salmonella sonnei. That's how you say that word. This one used to go by a different name. It used to be called Shigella sonnei, and in fact, in some sources, it's still called Shigella. This is a frustrating thing that we do in science. Sometimes we rename things, and it's frustrating because I know in my personal professional career, this organism has undergone like 4 name changes. And it's because different scientists believe, oh no, it's a salmonella. It's genetically closer to salmonella. And other scientists say, no, it's genetically closer to Shigella. So they fight. And the name goes back and forth. So depending on the source you read, it will either be called Salmonella sonnei or it will be called Shigella sonnei. And finally, we have our old friend Pseudomonas, Pseudomonas aeruginosa, also an intestinal organism, a fecal organism, but not a coliform. And just like all of these, Pseudomonas can be a pathogen from time to time. In fact, we call Pseudomonas an opportunistic pathogen because Pseudomonas takes advantage of situations when people are run down. That's all that term is really referring to. Pseudomonas can be very problematic in hospitals. Remember, we said in an earlier lab that Pseudomonas is often growing in the environment, especially in wet areas. And a lot of strains are not going to harm us, but if you get a patient into the hospital who is very compromised, and one of the most horrific examples of that is a burn patient. If you get a burn patient who is severely burned and they come in contact with Pseudomonas, the Pseudomonas is going to take advantage of the fact that that patient doesn't have a barrier anymore in their skin. And the Pseudomonas will start to grow and it will infect them. So it's opportunistic. It takes advantage of a situation where it might not be able to bother you and I because our skin is intact. Our immune system is perfectly healthy and we're not fighting anything else. It attacks patients who are already compromised and can cause pretty serious infections. All right, so those are our 5 organisms. Now you can see all 5 of these could be causing an intestinal infection, right? If I got a sample in to the laboratory and the physician noted on the sample that it came from a patient with a bad intestinal infection, the first thing I'm going to do is gram stain that organism. But look, these are all gram-negative rods. Lots of intestinal organisms are gram-negative. Not all of them, but a lot of them. The gram stain, in other words, isn't particularly helpful to help me identify it. I need other information as well. I'm going to record the gram stain. I want to know that. I want that piece of data, but I also need to do other tests so I know what organism I have. So today we're going to look at 5 different gram-negative rod intestinal organisms and see if we can tell them apart. So let's talk first about the carbohydrate fermentation test. Remember that this is a test that will tell us whether or not an organism can ferment a particular carbohydrate and whether or not it produces gas while it's doing that. Remember, we're always going to get an acid, but we may or may not get an alcohol. We may or may not get a gas. So this is what the media looks like when it's not inoculated. This is what you would get it as commercially. And we do usually purchase this media only because it's so convenient to buy it. You can see the broth has a nice pinkish red color, and that's because it has an indicator dye in it called phenol red. And like lots of our indicator dyes that we've seen so far, phenol red is going to turn yellow when there's an acid present. So if you put an organism in here and you let it grow and it ferments the sugar, whatever it is, the media is going to turn yellow. Now, the trick to the gas is that inside each one of these tubes, in the liquid, there's a second tube. It's called a Durham tube. It's just a little tiny narrow glass test tube. And what we do is we drop it into this larger test tube upside down. This right here shows you a Durham tube. So this is just an empty test tube right here. But see this right here? There's a second test tube in here. It's much smaller and it's upside down. This is the bottom of the tube. And this is the opening of the tube. So you can imagine if you dropped that little Durham tube in here, while this organism is fermenting the sugar in here, if it's making gas, some of that gas is going to get trapped in this tube. Remember, the gas is going to rise up. Some of it will escape through the broth, but some of it will get trapped right in this upside down tube. So when I check my carbohydrate fermentation tubes, I'm not only looking for a yellow color change, but I'm also looking to see if there's a gas bubble inside the Durham tube. It's a really ingenious little test if you think about it. Remember, we can put different carbohydrates into that tube. When you order your carbohydrate fermentation tubes, you can order whatever carbohydrate you want. If you want to look for lactose fermentation, you can have that added to the tube. If you want to test maltose, have that put in. If you want to test sucrose, put that in. You can order whatever you want, which is why, again, a lot of people opt to buy their fermentation medium. Now I have a tube right here of carbohydrate fermentation medium. And I think you can just see, maybe not. There's a Durham tube in here. Now it's hard to see. There's liquid in the Durham tube. So the Durham tube is in the bottom of this tube. Yeah, it's really hard to see because there's no gas in it. It's full of the same liquid broth that's in the rest of the tube. But take a look at this one. This one, I artificially put a little bit of air into the Durham tube. Now you can really see the Durham tube, right? And that's because it has a big gas bubble in it. See that? The little tube inside the red liquid has a big gas bubble in it. So that's what we're looking for. We're looking to see if there's gas in that little Durham tube after we allow the microbe to grow in the incubator overnight. So this is what the color change looks like. Yellow. Phenol red will turn yellow in the presence of acid. And there's a gas bubble in the Durham tube. Let me see if I can get you to see that. There, you can see it right there. There's a big gas bubble. About half of the Durham tube is full of gas. See the liquid line right there? Sorry, right there. You can see the liquid line inside the little Durham tube. It has been pushed down by gas. So this organism fermented the sugar and produced an acid, which is what turned it yellow. And it also produced a gas, which we caught in the Durham tube. Now, when you're preparing your carbohydrate fermentation tubes, you want to inoculate your media, with your best sterile technique. You want to hold the tube at an angle, and you want to keep the cover in your finger while you're inoculating. Put your cells in with your loop. And then you want to go ahead and just tighten your cap. The reason you want to tighten that cap is because you want to see if this organism can ferment. You want to see if it can ferment whatever sugar's in here. So I want to tighten my black cap because I don't want to let oxygen in overnight. There's still going to be some oxygen in here. So I would have trouble growing an obligate anaerobe, but any other kind of organism that doesn't find oxygen toxic, I can grow in here. And if I keep the cap closed, I can force it to ferment. All right, let's take a look at, again, what we expect to see in our Durham tubes. Normally, the tubes are red, and they have a Durham tube inside of them. And we always want to make sure that we start with the Durham tube full of liquid, because that will help us determine whether or not gas gets produced during incubation. We're going to add our cells into the tube. We're going to close the tube. The next day, when we come back in and check, take it out of the incubator, we're looking for the color change. Yellow means acid. That's what the A is for. You can see that there's still liquid all the way up this Durham tube. No gas. But this one looks different. There's yellow liquid right up to about here, but then there's a gas bubble up at the top. So this one produced acid and gas. That's what the AG is. Now, one thing we always do when we run carbohydrate fermentation test is we always run a control. We take one tube, and we make sure that it has nothing in it, no cells. And then we take another tube and we put the cells in. Here's my other tube right here. Both tubes go in the incubator. So I always run a control uninoculated tube, and I run my test. It's really important that you run a control. The control is going to look different every time, but you need the control because you have to be able to show that something other than the cells didn't cause a problem for you. In other words, if I pulled my control tube out of the incubator the next day, I know I didn't put any cells in here. If I pull it out of the incubator the next day and it's yellow, that means, there was a problem. There was a problem with my experiment. Something happened. Maybe it got contaminated. Maybe the indicator dye isn't working. Phenol red and other indicator dyes get old. And sometimes they turn color, not because there's an acid in the tube, but because the indicator dye isn't working correctly. So if I run a control tube with no cells in it and I get a color change, then I can't trust the tube that I put the cells in, because obviously something went wrong. Something went wrong. You see what I'm saying? Always run an uninoculated control tube with your inoculated tube. You're going to expect your control tube to stay pink and have no gas in it. And as long as that's what you see in your control tube the next day, you can trust what's happening in your experimental tube. Okay, I put this slide in. I just wanted to point out that it is possible to make your own carbohydrate fermentation tubes. And it's possible to sort of tweak the recipe of the broth. Remember, we've been talking a lot about how fastidious organisms have very particular nutritional needs, and sometimes they're very hard to grow. And sometimes scientists add additional ingredients into carbohydrate broth because they want to test for something very particular, or they want to grow a particularly fastidious organism. There's no problem with making your own broth, but I wanted to point out that sometimes when you make your own broth, depending on what you put in it, your starting color can be different. Notice how this starting color is not red. This is the red of the commercial medium. This still has phenol red in it. It's just that it has other ingredients in it too. And it has turned the media an orangey color to start with. It's important to know what your starting color is.
Can I ask you a question real quick?
Yeah.
Is anybody else having audio problems? Because I'm missing like 1/3 of what you're saying.
Oh, I'm sorry. Is anybody else having audio issues?
No, I'm not.
I'm not. Me neither.
I'm sorry about that.
That's fine. I might have to watch this like in the morning or something, because I literally can't hear almost half of what you're saying,
Oh,
and
I'm so sorry.
I can't figure out what your I can't lip read. So
Yeah, or the other thing you could do if you want is you could try logging out and then logging back in.
okay, I'll do that first.
You know, the mysteries of computers, right? I can't help you there, but otherwise, yeah, feel free if you need to step out and watch the recording. So as I was saying, you just want to make sure that you know what your starting color is, because you want to be able to know if it changes. That's what you're looking for. You're looking for a color change. And the phenol red will go to yellow if the pH becomes acidic. That's what you're going to see. And you can see that in these tubes. This tube is the uninoculated control. These 3 tubes had microbes put in. So here's E. coli. Here's, again, Salmonella sonnei, or maybe Shigella, depending on your reference. And here's Pseudomonas. These are 3 of the organisms that we're using today. And if you look, you've got one tube over here that looks just like the control, right? Just like it. It's still orange. There's no gas in the Durham tube. So nothing has happened in here. The Pseudomonas did not ferment the sugar in here. But look at these 2. The S. sonnei, it fermented just fine. It turned yellow. It produced an acid, and it turned the tube yellow. There's no gas in the Durham tube, though. So this would be acid, no gas. This one is acid and gas. This is E. coli. So we get a couple of pieces of information. Whether or not it turned yellow tells us if it fermented the sugar and produced acid, and whether or not there's gas is a visual in that Durham tube. All right, so here's our sort of official results for our organisms. Now, this is a graphic that came out of a textbook. I don't have actual pictures for you from what I made, but this is a comparison of glucose, lactose, and sucrose. So essentially what they did was they took E. coli, one of our organisms, Klebsiella Pneumoniae, one of our other organisms, and they ran them head to head. Again, these are 2 possibilities for somebody who has an intestinal infection, and both of these are coliforms. So we put them into carbohydrate fermentation tubes, incubated them overnight. Here's the control tube right here on the left. So this is the starting, again, the starting carbohydrate media. And the first sugar they put in was lactose. Then they inoculated one tube with E. coli and they inoculated another tube with Klebsiella, put them in the incubator. Then they took some more media and they put sucrose in and set up a tube for E. coli and a tube for Klebsiella. And then lastly, they did plain old glucose. Okay. So let's take a look down at the bottom of this image. I know it's hard to see, but I'll read it out to you. E. coli is in the top row and Klebsiella pneumoniae is in the bottom row. In the glucose tube, we saw acid and gas for each one of these organisms. So again, yellow media, bubble in the tube. So both E. coli and Klebsiella were able to produce acid and gas from glucose. Not all bacteria are going to do that. Lactose tube, same thing. Sucrose tube, same thing. So here we have two organisms that are pretty closely related to each other, right? Gram-negative rods, both coliforms. They can ferment 1, 2, 3 sugars and produce gas each time. Okay. Now, we ran a very similar test. Again, we tested glucose, lactose, and sucrose, but this time just on that Shigella sonii. Here's the glucose tube. A day after inoculation, so 24 hours in the incubator. Here's the lactose tube. Here's the sucrose tube. No reaction in here. No reaction in here. In glucose, fermentation occurred and gas was produced. So Shigella looks very different from E. coli and from Klebsiella. So just from this one test, and those 3 sugars that we tested, I can't really tell the E. coli and the Klebsiella apart yet, can I? But I can tell E. coli or Klebsiella apart from that Shigella by running those tests, by running those 3 sugars. Let's take a look at some other ones. Here's our old friend Pseudomonas. Here's the glucose tube. Here's the lactose tube. Here's the sucrose tube. No reaction, no fermentation. No reaction, no fermentation. Fermentation of the glucose. But no gas produced. No gas produced. Shigella and Pseudomonas look very similar, don't they? But they're not exactly similar. That's why that Durham tube is so important. Pseudomonas can ferment glucose. So can the Shigella. They can both do it. Neither one of them can ferment lactose. Neither one of them can ferment sucrose. But they both ferment glucose. We can still tell them apart. We can tell them apart because only the Shigella is going to make gas while it ferments the glucose. So they're both fermenting the same sugar, but they're using a different pathway. They're using a different form of fermentation. That's what I mean when I say there are many different fermentation pathways. 2 different microbes can ferment the same sugar, but they can ferment it differently. In other words, they can make a different product from that sugar. It might be an acid. Well, it will be an acid. It might also be an alcohol. It might also be a gas. So that little Durham tube in there is really important because sometimes that's the only distinction we have between two organisms. They both can ferment the sugar, but only one of them maybe can produce a gas while it's doing it. So there's a difference between saying an organism can ferment glucose and knowing the pathway that it's using to ferment the glucose. So this is a table of the results that we got from 4 of our organisms. You'll notice that Enterobacter is not on here yet. I'm saving the Enterobacter for another one of our tests. We tested all 4 of these organisms using carbohydrate fermentation tubes. We put glucose in one tube, we put lactose in one tube, we put sucrose in one tube. E. coli and Klebsiella pneumoniae were able to ferment all 3 sugars. Now remember what that means. That means that these organisms were able to take sucrose, for example, and using an enzyme, break it apart to set glucose free, because sucrose contains glucose and fructose bound together. So the reason that it's able to ferment the sucrose is because it has an enzyme that can break down the sucrose. And then the glucose molecule inside the sucrose is going to be fermented. It's going to be broken down into pyruvate and then into some kind of product molecule. One of those products is an acid. And for these 2 organisms, one of those products is also a gas. Same thing for the lactose. Break the lactose apart first into glucose and galactose. And then the glucose gets fermented into pyruvate. And the pyruvate gets turned into, for these organisms anyway, acid and gas. And here's just the glucose. So we sort of skipped one of the steps and just put the glucose in the tube. And yes, both can ferment the glucose. So these tests would not be helpful in helping us distinguish these 2 organisms. But it sure helps us distinguishing these other organisms, because the Salmonella or Shigella sonnei and the Pseudomonas, their results look very different. They can't ferment these more complex sugars. They can ferment glucose, though. That means they don't have the enzyme that's necessary to break the sucrose into glucose and fructose. They don't have the enzyme needed to break the lactose into glucose and galactose. They just don't have those enzymes. So they weren't able to get the glucose out. They weren't able to ferment. The Shigella made an acid and a gas. The Pseudomonas just made the acid. So we have a difference there that we could use in the lab to help us differentiate between those 2 organisms. Any questions? Mary, do you have a question? Any questions at all? It's another test that we can run in the laboratory, this carbohydrate fermentation test. Remember, the OF test, oxidative fermentative, that is going to tell us if an organism can ferment, because we're going to put glucose into that media. But this test not only tells us if it can ferment, but what it can ferment. Yeah, what it can ferment, because that's a real characteristic of different species of bacteria. All right, let's move on from here a little bit. We're going to move on next to this Mrvp test. Now remember, this stands for methyl red Voges Proskauer. The methyl red is just the name of an indicator dye. And the Voges Proskauer is just the name of the scientists who developed this test. These two tests are run together, one in each separate tube. So for example, you're seeing right here, it says negative methyl red, positive methyl red. So we do two tubes together for this test. One of them is going to be the methyl red test, and the other one is going to be the Voges Proskauer test. And the reason we run them together is because they're connected to each other. This media that is used for both of these tests contains glucose in it. It also contains an indicator dye called methyl red. Now, it's important to know that methyl red and phenol red are not the same thing. Phenol red starts pink, turns yellow. Methyl red is different. In the methyl red test, we're going to go from yellow to red. It's going to start yellow. And if we have a reaction, it's going to turn red. So I'll mention here that it's really important that you know indicator dyes, that you know how they work, because each indicator dye has a different color associated with different pHs. So it's an important thing to note that different indicator dyes are going to turn different colors under different conditions of pH. All right. So methyl red starts off, or I should say the methyl red medium for the test starts off yellow. We're going to get a change to that yellow color if an acid is produced, just like we've seen before. But we're not just going to get one possible change in color. We're going to get a range of different colors. If the tube turns red, and I mean cherry red, we know that the organism produced an acid, and that acid has a relatively low pH. It's less than about 4.4. Now, another thing we might see is that it turns more of a orangey color. And if it turns orange, we still have acid produced, but it's less acidic. And then you can get an even lighter color if it's even less acidic. So you get a range of color changes based upon how strong or weak the acid is. Voges Proskauer is different. Voges Proskauer media starts off when it's uninoculated as this sort of gross looking beige kind of green color. It's not the same as this media. So it has a starting color that's very different. Now, when you put cells into this and you grow them up overnight, you're going to look for a color change, but only at the top of the tube. You're going to look for this reddish colored ring. And it's not quite as simple as the other tests that we've looked at so far, because it turns out when you take the VP tube, Voges Proskauer, out of the incubator, you have to add a chemical to it. So you put the cells in it, you put it in the incubator overnight, take it out of the incubator, and then you put a couple of drops of this reagent into the tube, and you look for that red ring to form. So it's a little more complex of a test than anything we've seen so far. Remember, I also said that we're going to test, we're going to look to see if the organism can take the acid that it has produced through fermentation and convert it into this molecule called acetoin. So that's what we can also use this test for. So take a look at this slide. This slide compares an Mrvp test for E. coli with an Mrvp test for Enterobacter aerogenes. This is that last organism that was on our list today. E. coli and Enterobacter aerogenes are easy to mix up with each other in the laboratory because they have a lot of characteristics in common. They're both gram-negative rods. They're both fecal organisms or intestinal organisms. They're both coliforms, which means they can both ferment lactose. So you need a test that can distinguish them because they can both cause disease. The Mrvp test is a great way to distinguish these two organisms from each other because they literally have opposite results. So these tubes over here, this is the Enterobacter aerogenes in tube A. This is the E. coli in tube B. So I put the cells in to that, again, that first tube, and I put it in the incubator, and then I come back and I check. And then I put the E. coli in, and I come back and I check. At the end of the 24-hour period, the Enterobacter aerogenes tube with the methyl red test is still going to be yellow. In other words, we get a negative methyl red test. You can see the MR right here for Enterobacter aerogenes. But for E. coli, which is in the B tube, we get a positive test. So negative methyl red for Enterobacter, positive methyl red for E. coli. And in the VP tubes, we see exactly the opposite. Enterobacter aerogenes will give you a positive VP test. That's the red ring I was telling you about. And E. coli will give you a negative VP test, still that brownish, greenish looking media, not a very pretty media. So Mrvp can be useful for distinguishing these two particular coliforms. It's also useful for other organisms, for identifying other organisms, but it's just particularly helpful with these two because they're both coliforms and they have opposite results. One has a positive MR and a negative VP, and one has a negative MR and a positive VP. So we're looking for just the color change in the MR from yellow, you start yellow, to red. If it's fermenting and producing an acid, it's going to turn it red. The VP is a little bit different. The VP, when it comes out of the incubator, you got to add a reagent to it and you got to look for that red ring. That's a positive test. If you get no red ring, that's a negative test. So again, this is just a summary of what I just said. E. coli has a positive MR. It's going to turn the media from yellow to red. But it has a negative VP, no red ring. Enterobacter is just the opposite. Enterobacter aerogenes will produce a negative MR, yellow, and a positive VP, red ring. Questions? Okay. We have one more test to look at today, and it's this citrate test. Remember, citrate is not a carbohydrate. Citrate is an acid. It just happens that some organisms can ferment it. Citrate is produced in several different metabolic pathways as an intermediate molecule. And some organisms can take that citrate out of that pathway and ferment it to make ATP. Now I have a couple of, I don't have a lot of citrate test tubes to show you, but This is an uninoculated citrate test. The color isn't the best, and that's because it's been sitting for a little while, but you'll have to trust me, this media is green. It starts off green. Now, you'll notice that this is a slant. Can you see that? There's not liquid in here, that's solid. So this is agar. And it is, it's inside of the agar. Lots of ingredients have been added, including this molecule called citrate and an indicator dye. So I'm going to take my cells, using my best technique, I would spread my cells across this slanted area of the agar. I don't need to grow colonies. I don't need to do colony morphology with this tube. I don't need to count colonies with this tube. I just want to look for a color change. So I just need this little bit, this little stretch of agar that I can spread cells on, put it in the incubator, and see what happens overnight. Now, the fun thing about the citrate test is the color. Citrate, a positive citrate test turns a brilliant blue color. And that's what you're seeing here. Now, this is not a slant, this is broth. So this is a citrate broth that is a positive result. Look at that blue color. You can't miss this, in other words. It's a strong enough change in color that you will notice it. Now let's take a look at our organisms. So I often refer to this test as the citrate test, but the technical name for this test is the Simmons citrate test. Just like all the other culture media we've been talking about, you can make this or you can purchase it pre-made. In my experience, most people will make their own citrate agar because it's cheap. Now, this is what the agar looks like when it's not inoculated, just like I just showed you. It's a deep green. It's a dark green. When you spread cells on this agar and incubate it overnight, if those cells can ferment citrate, you're going to get a blue color the next day. And that's what you see here. You can make agar slants that are citrate agar, or you can make agar broth, citrate broth, not agar broth. You can make either one, depending on the organism you're trying to grow. You can see that when you use the slant, you don't necessarily see the whole slant turning blue, but you'll get enough blue. You'll know if you have a positive result. If you use the broth form, all of the broth is going to turn blue. And another thing some people do is they take citrate agar, and instead of making a slant, they pour it into a petri dish, and they just make a dish of citrate agar. Most people make it themselves. So they're going to make it, in whatever form they prefer to use, basically. So it's all about the color change with this. So again, I'm only using two of our organisms here, the E. coli and the Enterobacter aerogenes, because these two are kind of hard to tell apart. Here's what the uninoculated agar looks like on a plate. Again, it's that real dark sort of a green color. All I did was I divided this plate in half with a Sharpie marker. I spread Enterobacter aerogenes on one side. I spread E. coli on the other side, put it in the incubator overnight, and look at that plate. That's how stark of a color change you're going to see with this test. The Enterobacter fermented the citrate and turned that medium, that agar medium, this blue color. The E. coli can't do it. The E. coli cannot ferment citrate. So again, I have another way to tell these two closely related organisms apart from each other. And that's what I put in this table. E. coli can't ferment citrate, so the agar stayed green. That's a negative test. Enterobacter aerogenes can ferment citrate. The agar turned blue. That's a positive test. So, different carbohydrates, different tests. The idea is the same. I'm gathering data about an organism, and I'm using different tests to ask slightly different questions. Once I have my results for each organism, once I gather all of the results, I know a lot about that organism. I know different sugars that it can ferment, and I can then go into a reference table and I can try to identify the organism based on those results. Now, there are organisms that share things in common. Like we saw today, Enterobacter and E. coli share a lot of things in common, but there are a couple of tests that will distinguish them. The Klebsiella and the E. coli, they share a lot in common, but there are some tests that will distinguish them. Salmonella and Pseudomonas, they share a lot in common, but there are some tests that will distinguish them. So that's what we're doing in the micro lab, is we're running these tests. We're running these tests on these unknown microbes, trying to get data so we can identify them. Who has questions? So remember, in a couple of weeks, we're almost done talking about the kinds of diagnostic tests that are most commonly used in the lab. We're going to talk about protein next, and tests we can use regarding protein metabolism. And then we finish up talking about a couple of tests that are more directly related to anaerobic respiration. And then once we finish talking about those tests, now you'll know a lot about a lot of different tests. And as your second assignment in here, I'm going to give you some data. I'm going to tell you of a story about a microbe that was isolated somewhere, either out in the world or out of somebody's body. And you're going to be the technician in the micro lab. You don't have to actually perform the tests. I'm just going to give you the results of the tests. And you're also going to have access to some reference tables that I'm going to give you and some websites you can visit. And using that source material, you're going to try to figure out what you have based on all those characteristics. So you'll have a chance to practice. You'll have a chance to practice using all of these tests by identifying an unknown microbe. Okay. No questions. All right, everybody. That's all I have for you tonight. I'm going to let you go.
Thank you.
If you need anything from me, don't hesitate to get in touch. And we will see you next time.
Good night.
Have a good evening.